Hello everybody, today's GC meeting's topic is identification of small molecule SR9009 that activates NRF2 to counteract cellular senescence. This picture shows the abstract aging-related secondary phenotype is a prominent feature of aging. Accumulation of SASP factors leads to pro-inflammatory response associated with chronic disease. Inhibition of aging and SASP is a strategy to prevent or control aging-related disease. It was further found that SR9009 effectively prevented cell senescence and inhibited SASP in both radiation-induced and oncogene-induced mouse aging models, thereby alleviating immune cell infiltration. They found that SR9009 inhibits cellular senescence through the NRF2 pathway in vitro and in vivo, and activation of NRF2 may be a novel therapeutic strategy to prevent cellular senescence. This picture shows the background about the SASP. Cellular senescence occurs in damaged cells and prevents them from proliferating in the organism. Cell damage itself doesn't directly lead to obvious signs of aging, but when the damage accumulation and reaches a certain limit, cells stop proliferation and cause visible to the naked eye tissue weakness and physical aging phenotype. SASP composition of different biological activities showed that induced it interact with other cells and form a mechanism to adjust local microenvironment, may cause or exacerbate aging-related disease. SR9009 is an agonist for the synthesis of the clock protein river B. NR1D1, NR1D2, also known as river B, is an important component of the circadian clock involved in tumor genesis, metabolism, and inflammation. SR9009 and NR1D1, NR1D2 specific agonists reduce body weight in obese mice by regulating circadian clock and metabolism. In this study, SR9009 was found to be a promising reliever that reduces ROS levels in and inhibits DNA damage and SASP by activating the NRF2 pathway. And I summarize the general idea of this article as the picture shows. In this study, the first part, they used human dermal fibroblast. These cells I will call HDF later. These cells have typical senescence-related phenotypes and typical choice for researching cell senescence. They tested the activity of beta-galactosidase, which is the marker of the cell senescence, and I will call the beta-galactosidase beta-gal later. Rapamycin was used as positive control in their study. They used doxorubicin induced cellular senescence. I will call the doxorubicin dox later. We can see when they use SR9009, the content of the beta-gal was decreased. Quantification of the proliferation rate by Ki67 staining confirmed that SR9009 increased the replication activity of HDF cells. The marker H3K9 ME3 was decreased in senescence cells and increased after SR9009 treatment. Then they did the Western blot to test some protein expression after treatment with SR9009. We can see that the protein expression levels of DNA damaging factors and the SASP regulatory factors decreased after administration. Then they did the RNA-seq. They set 4 groups. The first one is the control, and the second one is the control, give SR9009. The third group is the DOX, and the fourth group was the DOX at SR9009. These are genes for SASP-related factors. We can see the SASP genes downregulated after treatment with SR. They enriched the pathway where the downregulated gene was located. This gene enriched about aging pathway. They did the qPCR to test these genes in their RNA-seq data. When treated with SR, these SASP genes were downregulated. They get the same result compared with the RNA-seq data. ELISA also confirmed that SR9009 reduced IL-1 alpha and IL-1 beta in HDF cells. This picture shows the MTT and the cell cycle analysis showed enhanced cell proliferation and inhibition of cell cycle arrest. The results were consistent with the anti-aging effect of SR9009. This is a little summary in the first part. Then the second part in this article, they found the SR was more effective than rapamycin in reducing SASP. They tested the beta-gal again after treated with rapamycin and SR. Rapamycin reduced the DNA damaging factors p-ATM, pp53. We can see the picture below. SR9009 significantly decreased the expression of DNA damaging factors pp65, p21, and p16 compared with the rapamycin in this picture shows. Then they did the qPCR test SASP genes both in SR and rapamycin treated. This result suggests that SR9009 is more effective than rapamycin in reducing SASP, especially in IL and CXCL. We can see the data here. Yeah. This is a summary in the second part. We can have a look. And the third part, they found SR9009 is independent of NR1D1 and NR1D2 in preventing aging-related phenotypes. Why they do this is because SR was previously reported as a synthetic NR1D1 and NR1D2 ligand. Whether increased NR1D1 expression could prevent the aging phenotype was tested. As shown in figure A and B, overexpression of NR1D1 didn't reduce the activity of beta-gal in HDF cells. But on the contrary, overexpression of NR1D1 increased the expression of SASP factor and aggravated the DNA damage response. We can see the picture C and D. This result is consistent with previous reports that SR9009 has an NR1D1 independent effect. Then they designed the shRNA to knock down the NR1D1 and NR1D2 to see the result. In this picture, we can see the knockdown of NR1D1 and NR1D2 by shRNA couldn't eliminate the inhibitory effect of SR9009 on SASP expression. The results showed that NR1D1 and NR1D2 were not involved in SR9009 mediated senescence delay. This picture is the summary in the third part in this article. We can have a look. Then the fourth part in their article, they found the anti-aging effect of SR9009 is mainly dependent on the NRF2 pathway. GO analysis of gene upregulated after SR9009 treatment revealed a significant increase in cellular response to reactive oxygen species, redox process, and regulation of the cell cycle. NRF2 is one of the major regulators of antioxidant response. It binds to AREIs and activates transcription of various antioxidant genes, which are thought to fight ROS and may be responsible for the decrease in ROS levels. NRF2 protein levels in HDFS was significantly increased after SR9009 treatment. However, NRF2 mRNA expression level was slightly decreased, but not significantly. It speculated that the change of the NRF2 protein expression level may be the effect of post-transcription rather than transcriptional regulation. We can see in this. CHX is a protein senescence inhibitor was added to SR9009 treated HDF cells for 0, 20, and 40 minutes. After CHX treatment, NRF2 protein level in SR9009 treated cells decreased gradually. When they add SR, we can see in the same time the NRF2 protein expression was higher than control 1. Quantitative curve analysis showed the half-life of the NRF2 was about 30 minutes in control cells and 23 minutes in SR9009 treated cells, suggesting that SR9009 may regulate NRF2 protein expression after transcription. This experiment is about IP of NRF2 antibody showed that the expression level of Euclid NRF2 was slightly lower than SR9009 treatment, suggests that SR9009 inhibited the ubiquitination of NRF2. Then they knock out the NRF2 gene, and the result showed that SASP increased when they knock out the NRF2 gene. It means that NRF2 is essential in anti-aging. The NRF2 inhibitor ML385 significantly blocked the inhibitory effect of SR9009 on SASP factor and RS levels. It shows in Figure B to D. This result suggests that NRF2 is required for SR9009 to slow aging. In this picture, immune fluorescence analysis showed that nuclear NRF2 expression increased after SR treatment. And then SR9009 upregulated the expression of the HO1 and NQO1. Why they tested these two proteins is because they are the downstream target genes of NRF2. Then their interest focus on these two genes, HO1 and NQO1. When they overexpressed HO1, we can see the blue group compared with the green group. After overexpression the HO1, the SASP factors downregulated. They also overexpressed NQO1, but I didn't put this data here because this data is in their supplementary information. They do the same experiment. So this is also a summary in the first part in this article. We can have a look. This is the fifth part in this article. They found the decrease of ROS level is an important reason for the anti-aging effect of SR9009. Their previous study have shown that SR9009 induced apoptosis in cancer cells by inhibiting FAS and SCD1, inhibiting autophagy and de novo antipogenesis. Therefore, the expression of lipid-producing enzyme and core autophagy genes was detected. SR9009 slightly increased FAS expression without altering SCD1 and autophagy gene expression. In this picture, we can see SR9009 significantly reduced ROS levels. During chemotherapy-induced senescence, the The ROS inhibitor NAC alleviated senescence, SASP, and DNA damaging responses. We can see in the figure D to E. In conclusion, the decrease of the ROS have an inhibitory effect on senescence and SASP. And this is also the summary in this part. This is the last part they did experiment in vivo. SR9009 alleviates aging-related phenotypes in vivo. To explore whether SR9009 can inhibit aging and SASP in vivo, SR9009 treated mice were exposed to established dose of ionizing irradiation that induced DNA damage, senescence, and the previously described SASP. SR9009 treatment was continued every other day until 30 days after irradiation. The main focus is on the liver because it is thought to be a major organ associated with chronic inflammation that leads to a variety of disease, including fatty liver disease, like diabetes. SR9009 reduced beta-gal and SASP factors, including IL-1 alpha and IL-1 beta, IL-6, and CXCL1 in liver tissue. The result was the same as HDF cells in vitro. SR treatment reduced the accumulation of the T-cell marker CD3 and the macrophage marker MAC2, as indicated by immunohistochemical staining. This is shows in the red arrow, and they also use the qPCR2 analysis. This picture shows that SR9009 significantly reduced DNA damage response factors like PATM and gamma H2AX, upregulated the expression levels of NRF2, NQ1, and HO1 in liver. This picture shows that SR9009 can reduce level ROS level. And they also did the C. elegans experiment. The SR also can extend the lifespan and reduce the ROS levels in C. elegans. This is the summary in the last part in this article. Their in vivo experiment. They used the mouse model and the C. elegans model. So this is the all summary in this article. SR9009 can inhibit cellular senescence and attenuate senescence, DNA damage, and SASP by reducing ROS levels through the NRF2 pathway, independently of NR1D1 and NR1D2. SR9009 increased NRF2 expression and nuclear translocation. Both NRF2 activator and overexpression of NRF2 targeted genes NQ01 and NHO1 can effectively reduce aging and DNA damage. NRF2 accumulation occurs at the post-transcriptional level, but not at the transcriptional level. SR9009 also inhibited senescence, DNA damage, and SASP in IMR19 fibroblast. SR9009 was found to inhibit senescence, DNA damage, and SASP in vivo in a model of air-induced senescence. Release of SASP into the tissue's microenvironment can cause chronic inflammation. SR9009 can effectively reduce immune cell infiltration, suggesting that SR9009 can reduce chronic inflammation in vivo. May subsequently alleviate chronic inflammatory-related human diseases, namely fatty liver disease, diabetes. However, it needs to be validated in other vivo models of aging or chronic diseases, including aged mice, premature aging mice, diabetes, fatty liver. Inclusion SR9009 can inhibit aging, DNA damage, and SASP in vitro and in vivo by reducing ROS levels through activation of the NRF2 pathway.
Therefore, the studies are needed to clarify whether SR9009 can treat chronic inflammatory diseases. This is all. Thank you.